Okay, good afternoon, everybody. I'm very pleased to present today's seminar related with the influence of hydric hotspots on landscape-scale biogeochemical processes, conducted by Dr. Stephen Hall. So let me tell you a little bit about Dr. Stephen Hall. So he received a bachelor degree in botany, a master's degree and a master's degree in environmental and resources from the University of Wisconsin. His PhD studies was in ecosystem science at the University of California, Berkeley. Before coming to Iowa State, he did a postdoc at the University of Utah in the Global Change and Sustainability Center. Dr. Hall is a biogeochemist and ecosystem ecologist working in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. His research focuses on understanding the coupled biological and geochemical processes that control organic matter decomposition, nutrient cycling, and greenhouse gas production in soils. Last year, Dr. Hall's lab was awarded with the Center for Global and Regional Environmental Research grant for soil carbon cycling in Iowa agroecosystems, as well as an EPA grant to study environmental impacts on farm and restore pothole wetlands. So please join me to welcome him and for this presentation.
Great. Well, thank you, Carolina, for the very kind introduction, and thanks to Dr. Laird for the invitation. So I think most of us in this region are really intuitively aware of the presence of hydric soils at the landscape scale, right? Just driving around or walking around at your field site, right? Especially in the spring, you cannot avoid getting your feet wet. Well, I want you to think about the perspective of the microbial communities that are living in these systems, right? It's an entirely different world. If you are an aerobe versus an obligate anaerobe, and you're experiencing this spatial and temporal gradient of moisture availability. And I think we really need to sort of dial down our thinking about hydric soils to a microbial perspective. And so we can think about spatial scales of soil aggregates like we might have in our local mollisols. And we can think about these spatial scales relative to the spatial scales of microbial consortia, right, that are living within those biofilms, right? We have potentially 3 to 4 orders of magnitude difference. And we've known for decades now of the importance of fine-scale gradients in oxygen availability that we find within these aggregates. And so this is a classic paper by Sexton et al. It was actually conducted on a mollisol from eastern Iowa, where they isolated aggregates that were incubated under a bulk aerobic atmosphere, and they measured these oxygen profiles with microelectrodes, right? And so what they found is, over the scale of millimeters, right, from this, the aggregate exterior to the interior, right, we have these really steep oxygen gradients ranging from near atmospheric down to 0. And these gradients really emerge as interactions between the physical constraints on oxygen supply through the porous medium and the biological oxygen demand, right, both from the roots in the micropores and macropores, as well as the microbes that we might find living within these aggregated systems. And so just thinking of these spatial scales, we can quickly conceive of a diversity of other microbial metabolic processes that might actually become important at the ecosystem scale. And so traditionally in terrestrial soils, we tend to think about aerobic metabolism, especially in the context of organic matter decomposition, right? Where microbial communities are oxidizing complex or simpler organic material and oxidizing it to CO2, coupled to the reduction of oxygen to water. But as we saw within a spatially heterogeneous system, we can really quickly start depleting oxygen and moving down our kind of classical redox ladder. And we tend to think of denitrification as being episodically important, right, in our upland systems. But I want to highlight the potential importance of these other redox couples that are potentially available to this microbial community, right? Manganese oxides, iron oxide, sulfate, humics, and finally, methanogenesis. And so today I'm going to give you a couple of examples of potential insights that we can get by considering these anaerobic metabolic processes, even in what we oftentimes think of as relatively terrestrial systems, particularly focusing on this nitrate reduction couple as well as iron reduction. So basically the reduction of iron oxide phases to iron 2. And sort of my overall kind of take-home message that I hope to sort of support here is this idea that a better understanding of the distribution of these anaerobic metabolic processes can give us some insights into a number of the environmental processes that many of us care about, and many of us study. And first, as I briefly alluded to, organic matter decomposition, and especially the emission of greenhouse gases, carbon dioxide, nitrous oxide, and methane. There are a number of ways that anaerobiosis intersects with nutrient availability and losses, and as you might imagine, especially nitrate, but also phosphate and base cations. And I'll provide an example of this in a dramatically different system than we're familiar with on a day-to-day basis here. And finally, some of our more unusual processes relating to contaminants that are much less abundant but equally important. We know that process that converts elemental mercury to bioavailable forms is really closely related to anaerobic metabolisms, iron reduction, sulfate reduction. We know that the transport and mobilization of other redox-sensitive metals, things like arsenic and uranium, are also really intimately linked to some of these same overall themes that we'll talk about today. So I'll just provide a couple of case studies in this area from our group's recent work. And I guess the overall goal here is to understand some of these fundamental biogeochemical processes that occur at the scale of molecular to mineral organic complexes, how these ultimately scale up and how we can think about their distribution along soil profiles, among pedons, within a topographic sequence in a given landscape, and finally up to perhaps broader scales. So as I mentioned, really, I'd like to sort of think a little bit about where and when we might expect anaerobic microsites to occur in terrestrial ecosystems, and just a few examples of their potential relevance for global change biogeochemical processes. And so I'll use some case studies, both from some Puerto Rican oxisols from a tropical forest system, tropical rainforest system, where we've worked for a number of years, as well as some more recent studies in our own backyard. Okay, and so I think we can think about a number of approaches for trying to understand anaerobic biogeochemical processes in soils, right? We can take the approach of bringing out our microelectrodes and bringing out our oxygen sensors and trying to map these gradients over space or time, or we can take some other tacks. And so our group recently has looked at iron as a potential biogeochemical integrator of the redox environment. So we're using actually the solid phase as a reporter for both the recent and the long-term oxygen history of a given environment. And we can do this because there is a diverse suite of anaerobic bacteria, both facultative and obligate anaerobes, such as geobacter shown here, that are able to use a diverse set of electron donors, carbohydrates, potentially hydrogen, other electron donors, to reduce iron mineral phases to iron 2, and in the process, right, generating electron equivalents to drive their own metabolism. And so at any given time in the soil, we can actually quantify the abundance of iron oxide phases versus their reduced equivalents. And we can also track these dynamics as they evolve over time, because we know that much of the reduced iron in soils eventually becomes oxidized by the entry of molecular oxygen, right? And so we have this dynamic iron redox cycle where iron 3 goes to iron 2 and back again. And there's some really interesting consequences for ecosystem processes that result from this cycling. And another kind of useful aspect of iron cycling is the visual component, right? And so if we take a core, this is looking head on at a core from one of our oxisol sites, where we see this nice matrix essentially that's coated by nanoscale goethite iron oxide minerals, and we can see in these anaerobic microsites, right? These are dissolved, and it leaves behind this kaolinite matrix, which you see emerging as this gray color, right? And so we could see a nice visual imprint as well, oftentimes, of this iron redox cycle. And so I want to walk through, and I guess one of the points, though, is to be wary of color, right? And so the absence of evidence is not the evidence of absence. And just because we don't see glaying in a given system does not necessarily mean that iron redox cycling is not occurring. And so just to kind of illustrate this, here are a couple of profiles that I'm going to talk about today. So to the left, we have one of our oxisols. And so this is developed under a per-humid tropical vegetation. This site gets about 4 meters of rainfall per year. It's a very, very wet system. This is formed from quartz diorite parent material. And we see, okay, here's the sort of top of the profile. This actually extends for about 5 meters till we hit bedrock, ultimately. And I want you to hazard a guess as maybe where you think iron reduction might be most important in this profile. How many people say the BT or the plinthite horizon? Any hands in the audience? So you have to vote for one of these. Okay, we have one vote for the bottom. How about the BTG? So the little bit of glaying visible. Any hands? Okay, yes, a few hands. How about the A horizon? Folks in the back didn't vote. That's no fair. You guys should know the answer too. Yeah. Okay. So hold that. So how about the mollisol? So how about so again, this is only going down to about 80 centimeters. How many people say A2 horizon? Maybe A1. A few. Plow layer. Anyone voting for the plow layer? Okay, most people aren't voting. That's okay. So I'll give you the answer, and so I'll talk about the Puerto Rico profile first. And so this is really interesting. But so if we go out to this profile and we take subsamples and we throw these soils into test tubes with half molar HCl to prevent any further iron oxidation from occurring, we go back to the lab, we extract these, and we measure kind of these surface phases, these kind of the really short range order iron 3 as well as any iron 2. We see an exponential decrease in iron 2 with depth in this profile. And this is sampled under relatively well-drained conditions, despite the fact that these soils get so much rain. There are abundant macropores in the system. So we can actually get a very similar answer if we take these samples and we really allow the iron reducers to go to work. So we can incubate these samples under an anaerobic atmosphere, and we can see how much iron 2 they can produce. And again, we see orders of magnitude decline from the A horizon with depth. And so what's going on here? So we actually did some further factorial additions where we gave them different electron donors, additional iron phases of different composition, and the take-home message really seems to be that carbon is ultimately limiting iron reduction in this system. It's really sort of explaining why we see potentially high reduction capacity at the surface. Okay, I'm gonna skip this and actually go on. So let's think about sort of some soils in our own backyard. So this is our classic soil catena on the Des Moines lobe. Right, we have local topographic highs where we have these relatively well-drained, perhaps clarion series that's grading down slope into Nicolette, Webster, Canisteo, ultimately down into our topographic depressions, right, where we find our Okoboji soils. And so we can think about, well, where might we expect to see iron reduction, within and among these soil series. So what we find actually is really interesting. And so I'll walk you through this graph, and the overall take-home is relatively similar to what we found in the Oxisol. And so here on the left we have our clarion, so our more upslope soil series, again left to right, moving down slope into the depressions, and here we have 3 depth increments. So we have the plow layer, 20 to 60 and 60 to 100, which we've just binned here for simplicity. And we can take soils from these positions. We can flush them with nitrogen in jars in the lab, right? We can allow them to go to work and see, well, can they reduce iron? Well, not much happens if we don't give them exogenous carbon. And so these soils were collected following corn harvest, I guess a couple of years ago now, and we can see at this phase they appear to be carbon limited. We give them a little bit of glucose and they go to town. And actually these iron reduction rates are of similar order of magnitude as we saw in these oxisols, with really high availability of these reactive iron phases. And again, kind of the salient trend is that we see this dramatic decline with depth, which is not what we see if we're just using soil color as our index of iron reduction. Okay. So what about other potential redox couples in this system? Well, I'll highlight denitrification capacity, and we're going to come back to this later, but we actually see qualitatively similar trends as we saw with iron reduction. So we can move across, right, this catena from ridges down into depressions, and we can move down with depth in the profile. We see a little more topographic variation here, right? We see some evidence that denitrification there's basically more adept denitrifying communities in these depression systems than on the ridges, but again, they appear to be most capable in the surface. And again, in these assays, we're giving them excess electron donors, excess nitrate. So this is really assaying who's there and what are their potential rates for denitrification. And so I think these are sort of useful exercises maybe to think about, well, where and when might anaerobic microsites in these systems be most important, right? And so I just showed you two kind of examples of relatively humid systems, but I think it's worth thinking about the potential role of these anaerobic microsites even in surface horizons of soils that are not necessarily regularly saturated. And this idea that in these relatively humid environments, in many cases, oxygen demand or vice versa, carbon supply might actually be really important in terms of structuring the distribution of these anaerobic processes. Okay, so this is great. We have these sort of boutique metabolic processes that are happening in surface soil. So why do we care? So we're going to dive a little more into the nitty-gritty here. And so what are some of the consequences? I think in the case of iron cycling, there's some really interesting dynamics that happen when we reduce and we oxidize these phases. And this is particularly interesting because of the importance of interactions among iron oxide phases and soil organic matter. And so this is a recent figure from a paper by Cleaver et al., sort of sketching out the nature of some of these interactions. And so here, let's see, what do we have? We have these idealized ferrihydrite particles in soil. So these are these brown globs here, and we have organic matter that's represented by these yellow dots, and we have monomeric iron as these red dots. And so basically we can imagine many different structural configurations by which iron and organic matter are interacting, ranging from simple adsorption, where we have an iron mineral and the organic matter is just stuck on the surface, to much more sort of complex spatial arrangements where we have co-precipitates of organic matter and iron, where there's actually much more organic matter on a molar basis relative to the iron that's present. And I think these second kinds of mechanisms could potentially be important even in systems with relatively low iron abundance, right? So if you take our local mollisols, perhaps we have 1 to 2% of total soil mass is iron, and even a smaller fraction of this might be in these reactive iron oxide phases. But I think the take-home point is that these are serving as sort of a connective structure that's structuring the colloidal or aggregate environment, or generating these co-precipitates. They could actually be ecologically significant, with some interesting sort of wrinkles, both for cycling of nutrients and for cycling of organic matter. So I'm going to transition to a sort of a brief example here, looking at the relationships between iron cycling and cation availability in a tropical forest. And so this is a similar kind of a cartoon with what I was just showing you, right? We have some idealized iron phases here, kind of in orange. I guess the color got kind of washed out. But essentially, we can think about some organic matter, perhaps this embedded with these iron phases. And on that organic matter, right, we have cation exchange sites. When this organic matter, right, is exposed maybe to the surface, these cations might be available to interact with the soil solution, or perhaps these are actually embedded right, within this mineral or within an aggregate, and perhaps these cations aren't available to interact with the soil solution. And this becomes a really interesting problem if you're a plant that's growing on a soil that's several 100,000 years old and your cations have been markedly depleted, right? If even a small chunk of cations are actually occluded within these mineral phases, you might have a nutritional problem. And so we wanted to see whether perhaps reduction of these iron phases by microbes within microsites could potentially alter the availability of these cations in the solution. So again, we're working in the Luquillo experimental forest, which is on the island of Puerto Rico. This is a forest system that gets on the order of 4 meters of rainfall per year. These are highly weathered oxisols where the cations are depleted on the order of 90 to 99% relative to parent material. All of the primary minerals are essentially gone, right? We think of primary minerals in many cases as the dominant source of cations during weathering. And so it really remains to be seen, well, many of the cations that are left appear to be associated with secondary minerals, not necessarily in exchangeable pools. By what mechanisms could plants potentially get at these phases? Okay, so to sort of answer this, to see whether this iron reduction hypothesis might be plausible, went out to the landscape and sampled about 120 plots that are stratified by topographic position, maintaining a common vegetation type. And I'll show you some data from ridges, right, where we're actually able to sort of remove some of these effects of material transport and et cetera. And so what we find, if we just grab samples from around the landscape and we look at the variability in iron 2, again, we're doing this by taking samples in the field, immediately immersing them in hydrochloric acid to prevent oxidation of this iron. We can actually see really strong relationships between reduced iron, which is here on the x-axis, and 3 of our cations, especially potassium, but also calcium and magnesium. We can explain about 70% of the variation in potassium simply as a function of iron 2, or the variability of iron 2 among these samples. You might say, well, this is just correlations, right? This is probably covariating with something else. So we looked at every other factor we could think of: fine root biomass, soil organic matter, cation exchange capacity. The list goes on. And really, if we look at both iron 2 and iron 3, they turn out to be some of the best predictors of cation availability at the landscape scale. So we can extend this a little further, right? We can take some of these samples back to the lab. We can drive iron reduction by exposing them to a nitrogen headspace. And again, we see really similar trends, right? As we're increasing concentrations of iron 2, we see release of magnesium, calcium, and potassium. And this holds up, in several different extractions that we can subject these soils to. So I think sort of the implication is potentially that these microsite scale dynamics, in these surface soil horizons could potentially play an important role in terms of plant nutrition, not only for cations, but also phosphorus. And I won't get into phosphorus today. Okay, so that's sort of one extreme example in the humid tropics. So let's come back closer to home, and let's think about a system that's much poorer in terms of reactive iron phases, at least an order of magnitude lower total iron content in our local systems. But we still wanted to see whether some of these iron redox dynamics might be important in terms of understanding in this case, the dynamics of organic matter. And so here's another sort of a cartoon, right, where we have a mineral matrix. It's mostly clays, right, smectite, et cetera. But there's a small amount of these reactive iron phases that might play a disproportionately important role. Perhaps if we dissolve them, we can liberate some of this organic matter, either as colloids, alter the aggregate structure, release some of this carbon to solution. Okay, so that's sort of the hypothesis we set out to test. And we were really interested in this in the context of understanding how soil carbon cycling understands to moisture. And so, right, if you look in your soil science textbook, you'll probably see something like this, where we see a really sort of characteristic bell-shaped response of respiration to soil moisture, right? Really dry soils, microbes are desiccation limited, right? Really wet soils. We have physical constraints on oxygen supply, and there's perhaps a happy medium, right, where both physical and ecological factors are optimum for microbial respiration. Well, we wanted to sort of push this a little further and ask, well, what happens when we're actually altering the geochemical environment as a function of moisture, right? So if we're driving iron reduction under these wet conditions and we're releasing organic substrates to solution, could this actually alter this moisture response curve that we typically see? And so another wrinkle we're going to add here is actually using the stable isotope composition of organic matter to get at potential changes in carbon cycling that are associated with iron reduction. And so I think we live in it. So this is a brief digression here, but we live in a fantastic part of the world to study carbon cycling because we're in the land of C3 and C4 transitions, right? And so we can think about plants with these different photosynthetic pathways as imparting isotope tracers onto the carbon cycle, right? And so our sort of classic land use here, corn soybean rotation, we have corn, which is a C4 plant, right, with a characteristic carbon isotope ratio. And so if you're not familiar with isotopes, don't worry about this notation. Just know that these different photosynthetic pathways fundamentally alter the isotope composition of this organic material, and we can follow this through as the organic material is decomposed. And we can actually measure this in carbon dioxide that's produced by microbes, potentially methane, whatever aspect of the carbon cycle, we can follow it with isotopes. Okay, so we've got our C4 plants, our corn, as well as many of our prairie grasses, C3 plants, soybeans, many of our herbaceous plants in prairies, right? And so over time, in soils, we have a balance between these C3 and these C4 plant inputs. And so what we can do actually is we can perturb our system, we can drive iron reduction, and we can actually test this question of whether this fundamentally alters the sources of organic matter that are available to microorganisms. And so we've developed some methods to sort of make this process a little bit easier. So we've taken a tunable diode laser that's oftentimes used for atmospheric measurements, and we've sort of streamlined this for use in soil studies. So we can inject small discrete samples, or we've set up a variety of different kinds of apparatus to actually measure production of CO2 from soil cores on an automated basis. This is an experiment that Cheng Long in the back is currently working on. It's sort of an outgrowth of our longer term efforts. And so what we can do then is use these isotopes to get at the question of how shifts in moisture and potential redox status might alter carbon cycling. And so this is work by Wenjin, who's also in the back, so I'm going to share her work right now. This is a laboratory experiment where we're taking replicate surface soil samples from across our Des Moines lobe catena from 3 different soil series. And sort of as a proof of concept, we're going to expose them to 3 different moisture levels, ranging from near field capacity, actually just below field capacity, to just above field capacity, and saturated. So these soils were harvested immediately following corn harvest, and so we amended them with some corn stover just to kind of juice the microbial communities, give them some fresh carbon to work on. And so what happened? And so as you might imagine, right, when we increase moisture in these systems, we see redox potential rapidly decreasing. So redox potential is a sort of integrated measure of the soil environment. And so essentially, let's see, what are we showing here? Redox potential is in these squares. And so the saturated treatment is here in orange. And so we see this rapid drop in EH just within a couple of days. Our control just below field capacity, we see no change in EH. So accompanying this, right, as you might expect, we see an increase in reduced iron. So we see iron reduction after just 2 days. And it's worth noting that these are the 2 end member treatments. We also saw significant iron reduction in the treatment that was just slightly above field capacity, which I think is worth noting. Okay. So along with these increases in ion reduction, what do we see in terms of carbon dynamics? So we see a fairly dramatic increase in dissolved organic carbon. So again, this is our saturated treatment here in orange, and our field capacity controls down at the bottom. And so we've actually split this out using isotope composition into C4 and C3 derived organic carbon. I'm going to come back to the isotopes in just a second. Okay, so what do we find in terms of microbial respiration? So at the beginning of this experiment, so first I'll just show you the first 25 days here. We see sort of this classical moisture response that your textbooks might predict, and we see this hold across the three soils that we examine. So what do we find? That the saturated soil has the lowest CO2 production, the intermediate soil is in the middle, and the field capacity soil has the highest CO2 production. Okay, no surprise here. And so what's more interesting, right, is we take this incubation out further in time. We see that after we get to about 30 days, these respiration trends really start to converge. Even when we drain our saturated soil, nothing much happens. And at the end of the experiment, CO2 production is essentially similar among these different treatments. And obviously, right, we're forgetting half of the carbon cycle story, right? And so what we find actually is that after about 3 weeks, we see significant methane production, both from the intermediate, both from the saturated treatment as well as the treatment that is just slightly wetter than field capacity. And so we see at the end of the experiment, methane accounts for about 30% of the total carbon losses from these wetter soils. And as a consequence, when we add up both CO2 and methane, we see that our total carbon losses are about 150% of the control in these treatments that received more water than field capacity. All right, so we can split this up according to isotopes, right? We can split this into carbon that came from a C4 plant and a C3 plant. And so we'll start off with C4. Right, so this is, in this system, the C4 carbon is the newest carbon, as well as a substantial fraction of the legacy carbon in the system. Again, in the beginning, we see this sharp suppression of respiration from this new carbon, but fairly quickly, right, the picture becomes pretty muddled, and we don't have many differences among treatments. So what happened with C3? Well, here is where we see a really marked divergence as a function of moisture. And so as we head out over time, we see that losses of this C3 carbon are markedly accelerated in these overly wet soils. And again, it appears that the preferential loss of this C3 carbon is what's really driving this treatment response in these systems. And so essentially, if we can revisit this kind of classic moisture response function, right? And so we can look at relative water saturation, and we can look at relative losses in carbon amongst these soils, right? And so after just 3 weeks, we see this sort of classical, parabolic response. But as we move out further in time, we see the emergence of this phenomenon that really I think is best evidence is this may be driven by iron reduction and release of previously protected carbon. Okay. So, right, these are kind of toy examples in a sense, right? We know in many of our systems we're not going to keep them consistently saturated for months at a time. Really, that's an end member, right? So I think one of the questions is, well, how can we think about these processes, whether they're at microsites within a profile, or perhaps macrosites, maybe scales of kind of swampy depressions? How important might these phenomena be at the landscape scale? And that's really where our kind of ongoing work is focused. And so I'll share some quick vignettes for where we're going here, and try to finish up with time for questions. So this is a new project in collaboration with several folks in my department. So as well as elsewhere on campus. So we're trying to understand the role of spatial and temporal variation in moisture on greenhouse gas emissions at sort of both the field scale and at the catchment scales. And one actually approach we're taking is to assess the fluxes of greenhouse gases in tile drain outlets. So right, many of our catchments, right, all of the tiles are linked to a main, which is xing at a defined point. And so we can actually try to quantify, well, how much of these anaerobic processes might be reflected in this tile drain water. And so we're coupling those hydrologic exports with high-frequency measurements of greenhouse gas fluxes from the soil surface to try to get at the role of these topographic gradients and kind of event scale responses. And really critical, I think, to acknowledge is really the role of dynamic hydroperiods in these depressions. And so this is some data from collaborators in the Ag and Biosystem Engineering Department. This is Amy Kalita's group. And these different colored lines are showing different pothole depressions just locally to Ames, and just showing you the hydrograph from 2016. And what we see is tremendous diversity both within and among rainfall events in terms of the duration of inundation, which can occur for anywhere from hours to days to a couple of weeks in some cases. And we're trying to understand really how these metabolic processes are responding at these event scales. And so one method that we've developed to get at this is actually an autosampler that captures dissolved greenhouse gases at high frequency. And so we're actually equilibrating a gas headspace in our autosampler with the flowing water that's coming out of our tile main. We're trying to get really the pulse and to see, well, can we figure out when and where nitrous oxide exports might be important. And just to give you kind of an example of some of the dynamics we see, this is some data from last summer. This is from a tile drain near Ames. We have I'm plotting methane concentrations in this tile drainage water, relatively close to atmospheric prior to the rainfall event. And we see this slow upswing to on the order of 50 times, or 2 ppm atmospheric, so 25 times atmospheric methane concentrations coming out of this tile water, really trying to nail down where this methane is coming from and what are the event scale dynamics. Okay, so what about gases coming out of the soil profile? Well, what we're doing is actually trying to measure these dynamics across topographic gradients, ranging from these depressions, right, where we might expect to find these episodic anaerobic processes, ranging up to more well-drained ridge positions. And so this is work that's led by Nate Lawrence in the back, who's a PhD student in our group. And so we've developed these autosamplers that are able to tolerate this kind of unfriendly environment. And so this is a design for measuring soil to atmosphere greenhouse gas fluxes that can tolerate being wet. There's no electronic parts on the chamber itself. This is pneumatic. So what we're doing is pumping the gases that are produced to a nearby shed where we actually analyze them, and we control a whole array of these chambers that are distributed from depressions up into more well-drained areas. So here's Nate, hard at work actually doing some other sampling associated with these chambers. And so ultimately we're moving these gases from the chambers to our fancy outhouse where we're actually measuring nitrous oxide and carbon dioxide. We'd love to get an analyzer for methane as well. Haven't got the money yet. And so essentially this work is going to be okay. So I'll share you a brief vignette from some of these data. So this is assessing the topographic variation in nitrous oxide fluxes following a precipitation event that was sufficient to flood the pothole. This is from, let's see, May 2017, I believe. And so what we have here are basically these colors are reflecting different topographic positions from the pothole in red and up to the ridge in blue. And we can see, I mean, obviously there's lots of spatial variability, but we can see trends towards much greater N2O production in this pothole, specifically following a rainfall event. Within about 24 hours, we're seeing this pulse of nitrous oxide that dissipates within a couple of days. Basically, the water is draining, and by the end of the week, we're essentially back to baseline. And so essentially what we can do is start integrating these data over space and time. And so our best sort of conservative estimate is that these hydric depressions have on the order of at least 50% greater cumulative nitrous oxide emission than uplands. And again, this is really consistent with a denitrification response to these individual rainfall events. So I want to wrap up and give you guys time for discussion. But I hope to have convinced you that we have some good evidence for the importance of a diverse suite of these anaerobic metabolic processes, even in what we might call terrestrial systems, and especially at their aquatic interfaces. So not only denitrification, right? We think about that really frequently, but also processes such as iron reduction, methanogenesis, right? We see methane coming out of our tile drains. What's its source, right? I think there's a lot of exciting questions we can answer. And I think the overall implication is that these, whether it's microsites in a tropical forest or maybe macrosites, maybe these topographic depressions where we pond water, these processes might have significant aggregate impacts at the ecosystem scale. So thanks to a variety of funders that have supported this work, as many of the collaborators and my group that have helped with this, as well as collaborators across campus that are involved with this pothole research. So especially Amy Kalita and Andy Van Lukey and Emily Heaton in your department, Michelle Superior, as well as Bill Crumpton and Matt Helmers. So hope there's some time for questions.
Now we open the forum for discussions.
Yeah, Brian. Yeah.
Thinking about your relationships between reduced iron and cations.
Yeah.
So what does it look like for the oxidized iron? Is it iron or is it?
It's the reverse, actually. That's a great point. And so you can do a multiple linear regression, and essentially you get a negative relationship between cation availability and iron 3. So it's sort of consistent with that occlusion. Yeah, that's a great point. I mean, really I feel like that's sort of a first blush to say, well, is this hypothesis plausible? And hopefully we can really sort of dive in with some more spatially explicit detail. Yeah, great. Yeah.
This work you showed at the end about the nitrous oxide emissions, have you tried doing that yet in fields that have the usual high doses of nitrogen fertilizer applied?
And this, that's a great point. And so this field is actually, it's ISU owned, and it's sort of the conventional management, right? It gets, 180 pounds per acre, is that right, Nate? So yeah.
Have you converted these results yet into pounds of nitrogen per acre? Or
I think the emission factor is 4% of applied nitrogen is coming off as N2O, and that's a conservative estimate. So that's that's a great point. And so this particular, so we can, I didn't want to show you the whole time series. We could spend a whole hour kind of walking through it, but essentially this is just after planting. There was the planting dose of fertilizer applied. I think it was UAN that went down with this, and essentially these kind of rainfall pulses get dampened as inorganic N availability goes down over the course of the season. But we still see the topographic effect. It's a little more muted. But yeah, so this is, I should have mentioned, this is really sort of, we tried to pick something that was sort of representative of kind of conventional ag.
Good.
Yeah.
Very good. I have an unrelated question. Earlier in the talk, you mentioned that looking at the cations, how the iron seemed to be correlating with some of the other cations. Your extractant was a 0.5 molar HCl.
Yes. Yeah.
Is there any evidence that was so caustic that it was by itself was burning up the...
Great point. And it's, and that it's actually, thankfully in this system, it's not caustic enough to really solubilize more than just the surface phase. Oh. And we actually compared that with, the reason we're using the half molar HCl is so we can essentially measure the reduced iron and the cations in the exact same sample. We loved it, but it strikingly agrees with, was ammonium chloride or one of the more conventional kind of cation extractions. We get very similar overall sort of exchangeable values at the landscape scale. So that's it's a great point. Yeah.
All right, thank you.
Yeah.
I was wondering about the N2 over time. Like they always talk about this big peak at the end of the winter.
We see that, and you could talk to Nate about that. Yeah, unfortunately, we have there's some engineering challenges, and I think we've mostly gotten those sorted out, but we missed part of the peak last year. We did see it. We did definitely see a peak in March or whenever we started measuring, but it's actually measuring at the moment, and we have not seen a peak yet. I don't think this winter. It hasn't thawed enough, maybe, or we're heading into the soybean phase, so there wasn't any fertilizer put down, so that could also be part of it. Yeah.
Earlier in your presentation, you showed some results for 3 different water contents. I think saturated water content.
Yeah.
I think something above field capacity.
Yeah.
And field capacity. Mine's just a technical definition. How do you define field capacity? What do you mean by that?
Yeah. Well, and so again, that's sort of how did we define it for this study? I think we quantified it in terms of drainage after, like 2 days after saturation, right? That's one metric, or is it like minus 33 kPa? I think is the other metric, right? And so I forget what we chose for this particular study. Yeah. But again, it's also relative. Like in a sense, this peak of this curve is sort of defined in terms of that. But you're right. I mean, that is a potentially ambiguous term. But I think you could define it in terms of water potential, perhaps most rigorously.
Well, another question.
Yeah.
Really good presentation.
Yeah. Oh, thank you.
Confirmation of that.
Thank you.
It seems to me that it's a really good combination of soil microbiology, soil chemistry, and soil physics.
Thanks.
And I assume that you're characterizing some of the microbes. You want to know which microbes are involved in some of the chemical reactions, and can you focus
well, that's I think
a lot on iron and chemistry types of things. But you didn't say much about the physics, and other than water content. You use water content, but I'm just curious at some of the micro scale sites. Are you characterizing aggregate sizes? Are you characterizing pore sizes? Or where the water is seems to me is really what's important.
Yes.
Not bulk water content, but where does it concentrate? Where is it relative to where some of the microbes and chemical reactions are going on? I'm just curious.
No, I agree.
How you're characterizing the soils.
And that's a weak area for me. So we've done aggregate size work in the Oxisols, right? We're just getting into this local work. So hey, if you'd like to work with us, that would be fantastic. So that's definitely a weakness of mine. I mean, I've done some of these classic simplistic kind of aggregate size fractionation methods, but yeah, I think that's an area where we could improve for sure.
In your temporal nitrous oxide response, landscape scale response data set. Okay. Do you have any feel for how much of the nitrate that's being reduced here is from mineralization of organic matter, from translocation, from in situ? I mean, where it's coming from? How much of it is just simply that you're transporting large amounts of nitrate from upslope into these potholes?
Exactly. I think that's probably a dominant mechanism. Our lysimeter data, I think, are consistent with that. So I didn't show that. Again, we can get into the details. But yeah, we have good evidence for surface and shallow subsurface flow that's really concentrating nitrate in these positions. So I think that's a pretty parsimonious explanation.
Well, it is the simple explanation. I'm wondering if it's more. I mean,
I'm sure it's more complicated than that. Yeah. Well, what we'd love to do actually is use the isotopomers of nitrous oxide to try to get at how much is coming from nitrification, how much is coming from denitrification, and that's really where we'd like to go next. So these chambers are designed so they don't isotopically fractionate, and so we've actually demonstrated that with CO2 isotopes that this particular chamber design is robust in that way. And so love to get someone to buy us an analyzer so we can do that. So, but you're right. I'm sure there's a lot of, I think, just a first order question. Well, what is the mass balance and how is it distributed over? Like, hey, I think we're happy to get at that, but I agree there's much more kind of compelling questions we can start answering after that.
Yeah, we have have time for one more question.
Yeah.
Stephen, in the experiment where you incubated soil samples and measured the change in pH over time. I think you did add glucose to those.
We actually did. We gave him a little bit of corn stover. Yeah.
And you measured EH. Did you also measure pH?
We did, and it's remarkably stable, which is satisfying in these pretty well buffered systems. Yeah. So that's sort of one attribute that's nicer working in these mollisols as opposed to the oxisols, right? We have much less of this covariation.
That's interesting. That's different from what we normally find, because usually when EH goes down, pH goes up.
Yeah, maybe in an unbuffered system. But so that's a really good question. Yeah.
And that has an impact on the solubility of the organic.
Yes. Yes. Oh, understood. Yeah. And so that's one actually I think advantage of our study is that there's enough buffering capacity in these soils that we can actually control for the effects of pH alone. And so that's been one question is how much of the DOC release is actually iron mediated versus pH.
So
and
one of these was can steel. So it was calcareous. So that would surely buffer.
Yes. Exactly. Yeah.
The other was Aquarium.
Yeah, and we've got the data. Yeah, and I don't know how long ago these were limed. So that's one question that it's obviously farmers are doing what they do on the landscape, and that was.
Did you measure the pH?
Oh yeah, we measured it. You could read the paper. Yeah. And over time. So it's all in the SI. But I think one final note is that we actually oftentimes see pH decreases under sustained reducing conditions that we actually are attributing to organic acids. And so so we also see the reverse, right? So okay, if you just look at your chemical equations, right, you might expect pH increase, but there's all of the interactions in the system that can change the story. Yeah.
Thank you very much.
Yeah, thank you. Thank you.
So you're all invited for